Hello everyone, this is Dr. Prithpal Singh and MD Pharmacology from CMC Ludhiana. And I had started my series of generic drugs versus branded drugs, giving the concepts of generic drugs in the last two sessions. To continue with this further, this session today, I'll be concluding the session of generic drugs and how we can categorize or how we can characterize which one is a better generic drug. And I would also be talking about the various myths and facts that are pertaining to the generic drug. So I hope that after the end of this session, I am able to clear some of your doubts. And if there is any query pertaining to this, you could get back to me in my chat box, and I'll be happy to answer those queries that you have put in. So when we start with generic drugs, as I was discussing, I just have a brief recap of what generic drugs are all about. So generic drugs as such are identical or bioequivalent to a brand name of drug in dosage, form, safety, strength, route of administration, quality, performance characteristic, and intended use. So these are intended to be interchangeably used with the original product. They are usually manufactured without a license. They are marketed after the expiry of patent and marketed either under a non-proprietor name or under a brand. This is the basic properties of a generic drug, and today we'll be talking about what are the other aspects that can be taken into consideration. Moving beyond this, we already had discussed about the clinical trial where I told you the patent life is somewhere around 20 years and around 10 to 12 years. If under optimal conditions, everything goes in order, 10 to 12 years are taken before the drug is approved for marketing surveillance. So ideally speaking, an ideal situation, the exclusivity right of a particular product by a company is hardly for 5 to 6 years. Now, the generic drugs underwent a major reshuffle with the Hatch-Waxman Amendment to this FFD&C Act 1984, and this is one of the most successful pieces of legislation that has been produced, leading to a creation of the generic industry, where you can easily see the amount of prescription of generic drug rising to by more than 44%. Now, this particular act, it gives the company the sole right to sell the drug till the patent is effect. And once the patent is over, the companies can go for an abbreviated new drug application for approval to the marketing product. Now, how is this abbreviated new drug development different from a new drug application? Because in a new drug application, the animal studies, clinical studies, and bioavailability studies tend to take a lot of time, whereas the top 5 steps are similar. But instead of having animal studies or clinical studies, we mainly have the bioequivalence studies that tend to have a major role in this particular preparation. And we have to ensure that the quality of generic product is good. So there is a 5-step review process. We have already talked about it, and the scientific literature should already be there, and this product should known to be safe for the purpose. Now, whatever happens is, whenever we are talking about this abbreviated new drug application, this is the application is reviewed whether it is acceptable or not. It is under 4 headings, that is for plant inspection, chemistry, labeling, and bioequivalence review. If there's any problem in any of these conditions, then this particular drug is not approved as a generic drug. Now, generic drugs are mainly meant to be used for the same active ingredient, same route of administration, same dosage form, same strength, and same condition of use, which is compared to the reference listed drug. So they should have the same as the brand name labeling, but may delete a portion of label protected by patent or exclusivity, and they may differ in expecient, the pharmacokinetic data, and how it was procured must be supplied. So chemistry involves the component and composition, manufacturing, batch formulation, description of facilities, specification and test, packaging, and stability. Manufacturing compliance, for this particular, the purpose is to assure the quality of marketed product. The mechanism is to go for surveillance of the product, the manufacturing testing, and assess the firm's compliance with good manufacturing processes. So once this particular drug is approved, it comes under the electronic orange book of the FDA, which is which tends to tell us which particular drug is approved. And this orange approved drug has two equivalence codes, that is a code of A and a code of B. A means that this particular drug is substitutable, whereas B means that you cannot substitute this particular drug with the original drug. Now, there are certain terms that are used for bioequivalence, and we tend to use the terms pharmaceutical equivalent, pharmaceutical alternatives, therapeutic equivalence, bioequivalence, and bioavailability. So, when the products are considered pharmaceutically equivalent, if they have the same ingredient, same dosage, same route of administration, and identical strength or concentration. Pharmaceutical alternatives are if they contain the same therapeutic moiety but have different salt, ester, or complexes of the same moiety and are in different dosage forms, then they are considered as pharmaceutical alternatives. Therapeutic equivalent is a drug product that is considered if it is pharmaceutically equivalent, bioequivalent, approved as safe and effective, adequately labeled, and manufactured in compliance with good manufacturing practice regulations. What is bioequivalence now? Bioequivalence is these are pharmaceutical equivalents whose rate and extent of absorption are not statistically different. This is the major term. Statistically different when administered to the patient or subject at the same molar dose under similar experimental conditions. Bioequivalence may be demonstrated through both in vivo and in vitro methods, and even through pharmacodynamic studies and comparative clinical trials. There is a strong clinical correlation. We tend to go for single dose pharmacokinetics, and we have the pharmacokinetic profile that is comparable to the healthy volunteers. Bioavailability as such is the rate and extent to which an active ingredient in the pharmaceutical equivalent becomes available to the site of drug action, and this is to measure the it is for measurement to reflect the rate and extent of absorption of a particular active ingredient inside the body. Now, as per the FDA guidelines, the generic drug manufacturing must demonstrate that drug is bioequivalent to the reference drug, and in order to have the FDA preference, you should have pharmacokinetic studies, pharmacodynamic studies, comparative clinical trial, and in vitro studies. And then you tend to monitor the area under the curve, which should be almost similar, like this is mentioned out here. The Cmax and Tmax should be comparable. Now, when we talk about the limits, the limit is considered plus minus 20%, so somewhere around 80 to 125%. Considering this is the reference drug that is there. We have two compounds that are intended to be used as generic drug. This is product A and this is product B. Now, what you find is the study bioequivalence of product A shoots beyond 125%. That means that this particular product is not bioequivalent. And a product A which has a level somewhere around up to 125% is bioequivalent. So anything, even if this was somewhere around 85%, it would have been an acceptable level of bioequivalent drug. So what happens in the pharmacokinetic studies when we talk about healthy volunteers versus patient? If two drugs perform the same in healthy volunteers, the assumption is made that they will perform the same way in the patients with the disease, except in case of some drugs that are potentially toxic. So if we take into consideration the toxicity, then both the drug would behave in a similar manner whether we give it in a healthy volunteer or in a patient when we want to elicit the pharmacokinetic studies. Now, this was what we had discussed previously. Now we'll continue with where we had left the sessions. If there's any doubt regarding these sessions, they are already there on my channel as generic drug 1 and generic drug 2. So today I'll be continuing further with the FDA coding of bioequivalent products. Now, once we have the drug that is available, how do we code them? So the first letter of this coding system is A or B, which I told you in the just prior to when I was talking about the recap. A is when this particular drug is substitutable. B is when it is not substitutable. So, and then after this, we have a second letter as per the FDA coding system, which indicates the additional information on the basis of FDA evaluation. So let us consider what are these second letter, that what would this second letter indicate? So when we talk about this first letter A, as I've been pointing out, So this particular drug is therapeutic equivalent to other pharmaceutical equivalent product, and it is substitutable. Whereas drug B, when we are talking about this, this is not therapeutically equivalent and it is also not pharmaceutically equivalent also. Then coming to the second letter that is there, supposedly the second letter is A. So if it is AA, that means that this particular drug is substitutable. Now what further? Beyond this, what does it stand for? It stands for that this particular product is in the conventional dosage form. The reference product has no bioequivalency problem, no bioequivalency test performed comparing innovator and generic drug. Oral dosage form must meet an acceptable level in vitro dissolution standard. Whereas if the second letter is B, that means the product is meeting the necessary bioequivalence requirement. The actual or potential bioequivalence problems have been resolved through appropriate scientific testing. When more than one reference products of the same strength exist, a number is added to the AB code 1, 2, or 3. When we have more than one reference product of the same compound, the number designated which generic product is bioequivalent to the reference product, that is what this particular number stands for. We have additional rating problems such as when we talk about a rating label as AN. So that means the drug product is powder is available as solution and powder for aerosolization, and it is marketed for use in any of the severe delivery system, whether it is as well as pharmaceutical equivalent and therapeutic equivalent also. AOA is an injectable oil solution which is both pharmaceutically equivalent and therapeutically equivalent to the active ingredient. AP is an injectable aqueous solution, and it might be sometimes an intravenous non-aqueous solution also. And this is therapeutic equivalent to the product. And AT is a topical product. So N stands for aerolization, O for oil solution, P for aqueous solution, injectable, aqueous or non-aqueous, and T is the topical product. Then we talk about the B-rated products, which are not substitutable. That means if the second letter is BB, that means the active ingredient and dosage form with the documented bioequivalence has certain problems and it needs to be clarified. BP is both active ingredient and dosage forms are with potential bioequivalence problems. So it has a documented and this is potential. BX is data from the FDA are insufficient to determine whether this particular drug could be therapeutic equivalent or not. Now, what could be the potential benefits of a generic drug? So the potential benefits are we have a better access to the needed medicine. Well-chosen generics make government or household spend less without loss of quality or safety. The 1998 study by US Congressional Budget Office said that the average generic medicine prescription price was less than 1/3. So this is a sufficient chunk of average price of a single source innovator brand product. Now let's talk about the price differences between various generic and branded products as per the country. So what you find is that in most of the European countries, let's talk about Belgium, Italy, Spain, or any other for that sake, France, the product price difference is somewhere around 25 to 35%. The major price difference is somewhere around UK and US, where it accounts for 50 to 80%. So then what is the other potential benefits? It has an impact on the price of non-generic through competition. Generics may be the only way to bring price competition to the market that is based on the product differentiation, patent protection, and heavy brand promotion to other prescribers and patients access to the needed medicine. Now let's talk about why they are less expensive. Now these particular drugs are less expensive not because they are inherently indifferent in composition from the patented drug, but they are less expensive because of the structure of the generic market. They are competitive, they are free from the IPR, and they are often without R&D, and particularly the marketing cost that goes into branded proprietary drug. We were talking about animal testing, clinical testing, all that is taken care of. Now let's talk about certain myths and facts that are associated with generic drugs. So coming to the myth 1, it is said that generic drugs are not as safe as the branded name drugs. So this could be partly true and partly false, depending upon the cost of the drug. A drug that has Sufficiently low cost obviously would not be safe. So, but when you talk about a drug that follows the norm as per the guidelines, then the generic drug uses the same ingredient, works in the same body, and has the same risk benefit profile. If the condition that lies for a generic drug is that provided it has fulfilled all the regulations that are there to label it as a generic drug. Talking about the myth two, generic drugs are not as potent as brand name drugs. So let's clear this out. The generic drugs have the same quality, strength, purity, and stability. Myth three is generic takes longer to act inside the body. This is not true. They deliver the same amount of active ingredient in the same time as the original product. Coming to myth four, brand name drugs are made in modern manufacturing facilities and sub-generics are often made in substandard facilities. No, that is not the case. Substandard facilities are not permitted by the FDA. And if you go through the social media and various news agencies, you will get a lot of news regarding how FDA is conducting various scrutiny checks and how some particular manufacturing companies are omitted or excluded out from the generic market of US and Europe. This brings to the end of this particular session on generic drugs. And this is the address of the contacting office of the generic drug. And if you want to have clear any doubt about this, this is the index that is there. So I hope, I believe, I hope, and I think that I have been able to clear some doubts, some myths, some facts about generic drugs. And repeatedly, I would highlight again on the same aspect that generic drugs are only and only safe if they fulfill all the regulatory requirements. Provided a drug fulfill all the regulatory requirements, then that particular generic drug can be considered safe. So I hope you have had a good, happy learning from my side. And if there are any queries and any problems, you can get back to me. I'll be happy to answer them. So till that time, have a good day and happy learning. Thank you so much for your patient hearing.